It's absolutely my honor to introduce the team at Lundquist. A pleasure to collaborate with them on bringing forward clinical trials for children here and now. I'd like to introduce Dr. Lynda Polgreen, Associate Professor at the Lundquist Institute and UCLA Harbor Medical Center. She's a pediatric endocrinologist with special interest in MPS diseases and immunology, and she will talk about the Anakinra study. And then we'll hear from Jonathan Acevedo, PhD candidate and Sanfilippo Fellow, along with Dr. Julie Tuccillo from Jazz Pharmaceuticals, talking about the potential of Cannabidiol in Sanfilippo. Dr. Polgreen.
All right. Thank you, Kara. I so appreciate this opportunity to come back again this year and talk about the final results from our clinical trial of Anakinra and Sanfilippo syndrome. Next slide, please. So first, starting off, why Anakinra? Why did we choose that to go forward with? And this is really based on the preclinical work of Dr. Brian Bigger, who looked in an animal model of MPS IIIA and had found that if he increased the interleukin-1 receptor antagonist levels in the mouse model in a few different ways, that this led to decreased neuroinflammation, increased memory, and decreased hyperactivity. And so based on that preclinical work, we thought about, well, how could we do this in humans? And there is a medication, Anakinra, that is a recombinant interleukin-1 receptor antagonist. So similar to how he genetically modified the animals to increase the levels of this, we could use this medication, Anakinra, to increase levels of interleukin-1 receptor antagonists. And this medication has been shown to cross the blood-brain barrier, which also made it a good option. In addition, Anakinra is approved by the FDA to treat other neuroinflammatory disorders and specifically a few other pediatric disorders. So the safety profile in the pediatric population was already well characterized. Next slide, please. So the study that we conducted was a phase 1/2 study, which means it was a first in human study. We were looking at safety and dosing and tolerability primarily, and collected data on efficacy as well. The study design is shown here. We had an 8-week observation period leading into the treatment period with Anakinra, which is the generic name, and Kineret being the brand name. And in the first 8 weeks, we started everybody on a same dose of 100 milligrams subcutaneous, so a shot just under the skin, kind of like an insulin injection. So 100 milligrams daily. And at 8 weeks and at 16 weeks, if the patient had not improved on the 2 areas that were most bothersome to the caregivers, your parents, then the dose was increased. It was doubled to 200 milligrams daily. And those 2 most bothersome symptoms were chosen at screening. So they had a list of options, and based on what was most important to the families, we dose adjusted that way. Once they had gotten to the 200 milligram dose, which actually everybody by week 16 had, they then continued on that dose through week 36 when the medication was stopped, and then we had an 8-week observational period at the end of the study. Next slide, please. So we enrolled 24 participants, and the characteristics of that group are shown here. The age range was 6 years to 26 years. The majority of the participants were white, non-Hispanic, and we had an equal distribution between males and females, so 50-50. 20 of the participants had MPS IIIA, 3 with MPS IIIB, and 1 with MPS IIIC. Next slide, please. And there's a lot of information on this figure. I won't go through all the details. This is just kind of a classic figure 1 of a clinical trial that shows you how many people were enrolled, what sort of treatment they received, the follow-up, and just the numbers for every stage of the study. And what I'll point out here is on the left side, the first 8 weeks of treatment was really kind of I call our primary analysis, our primary endpoint, because we designed the study to ensure that we had 20 who had received treatment by week 8. Once we had enrolled enough to have 20 completing week 8 treatment, then we stopped enrollment and followed those participants through to week 36 and 8 weeks after. So to get there, we enrolled 23 participants. 1 person was excluded due to persistent low white blood cells or neutropenia. And then an additional 3 had discontinued the intervention before week 8 to give us the 20 then at week 8. Those 20 continued through the trial, and we had some who had withdrawn to the point where by week 36, we had 13 that were still on treatment. 3 still continued in the study off treatment, but an additional 4 had been withdrawn and not followed up in the study, had been lost to follow up or just declined to continue. So the thing I'd like to point out here, though, that was brought up very clearly and nicely yesterday with the 2 sisters who are taking Anakinra, was that the daily injections are quite difficult for some patients and families, and there's some variability here for a variety of reasons. But we did have, as you can see here, 5 participants who stopped the treatment because of the intolerability of the daily injections. And so definitely one of the limitations of this approach is that it is given subcutaneously, and the dose that we found was needed, that 200 milligram dose, unfortunately is not available as a single injection. And so it requires 2 subcutaneous shots a day. We did not divide those out twice a day. We just give those once a day and to make it a little easier. So the whole process only has to occur once a day, but it is 2 shots. Next slide, please. So that's the tolerability. Now, in terms of safety, what the tables here show are the adverse events that occurred in at least 5% of participants. On the left side of your screen are the adverse events or AEs related to the injection. And so, as expected, based on experience in other populations, there were a lot of injection site reactions. Most commonly, redness at the injection site, which occurred in 74%, so 17 of the 23 participants who were treated. And there was also things like injection site swelling and bruising, none higher than what is expected or what was seen in other populations. Now, on the top there on the left, I'd just like to point out the participants with any SAEs. So SAEs are severe adverse events, and although there were a few severe adverse events during the study, none of those that were shown here, none of the ones that occurred were related to Anakinra, were instead related either to just an accidental injury or something else. On the right side, there we have the adverse events, the non-injection site reactions, non-injection site adverse events that occurred and on treatment are the first 2 columns, and after treatment, so those 8 weeks after they went off treatment on there on the right. So you can kind of compare the frequency of these AEs on treatment and off treatment, because we don't have any natural history data that we had available to us to compare that to. And what stands out here, again, it wasn't unexpected, but that we do see a bit of an increased prevalence of upper respiratory infections, of neutropenia, which is a decrease in the white blood cells, and in thrombocytopenia, which is a decrease in the platelets. And again, similar to what's been reported in other populations, none of them were clinically significant. And then the other difference, I guess, to point out here would be the COVID-19 symptoms. So we had 5 participants with COVID-19, all were mild infections while on Anakinra, and nobody reported developing COVID in the 8 weeks after treatment. Next slide, please. Okay, so now to get to the effects that we saw. One of the outcomes we looked at was the Sanfilippo Behavior Rating Scale. So this was developed by Elsa Shapiro and colleagues to specifically look at these 6, what they call clusters and domains in Sanfilippo syndrome. And so these figures are just the outcomes that they ask questions related to, going from the top left to right there: mood, anger, and aggression, social and emotional dysfunction, lack of fear. On the bottom, we've got executive dysfunction, movement disorders, and orality. And so how these figures here are set up, just to orient you to the data, we've got on the bottom, we call it the x-axis, the visit. So before the gray box are 8 weeks of observation. The data points you see within the gray boxes are the beginning to the end of treatment. And then from week 36 to week 44, on the right side of the shaded box, is the observation period, 8 weeks after they went off treatment. And a benefit or improvement in any of these outcomes is a decrease in the score. And so what we found here consistently is a trend that before treatment, so the left side of the shaded box, we see somewhat stable scores on all of these measures. And then once treatment is started, a consistent improvement through week 36 for all of the areas. Some that are statistically significant, which I've pointed out here, and the p-value is listed there, in all areas except lack of fear. And when we first saw that lack of fear, we thought, well, why would that be? Why would we see this such consistent trend of improvements and not anything in the lack of fear? And the more we thought about that, we decided that we think this is actually an expected finding, because there are data correlating the lack of fear, connecting it with atrophy of the amygdala. So amygdala, part of the brain that controls the fear mechanism, and this has been published by Protocol et al. But it's a loss of the brain tissue in that area. And we would not expect Anakinra, by decreasing inflammation, to restore that area of the brain. And so it's actually more reassuring to us and reaffirming, might be a better way to say it, that we did not see any changes in the lack of fear outcome, but we did see consistent improvements in all the others. Now, the other interesting thing to point out here is what happens when the patients went off Anakinra. So if you look at the right side of the shaded boxes, you see this consistent increase or rebound in symptoms back to baseline. And so when we look at the week 44 compared to week -8 or week 0 or day 1, there's no statistically significant difference between the 8 weeks post-treatment and before treatment. And so the benefits that we found here on the Sanfilippo behavior rating scale clearly were just during treatment, and then you go right back to baseline. They're not we call durable. They don't persist after the treatment has stopped. Next slide, please. And another outcome measure that we looked at to try to really individualize the treatment and the outcomes that we were looking at was what we called an individual clinical response. And so this was developed based on the caregiver study that Dr. O'Neill mentioned yesterday, that they had published, looking at what are the items that are most of concern to parents and caregivers. And so based on what was identified in that study, we listed out those items here, which you can see there's 15 of them, from sleep disturbances, hyperactivity, etc. And we asked each caregiver or parent at the beginning of the study, so at screening, to choose the 5 of those outcomes listed there that was most important to them, most stressful, most significant to them and their families and their children. And then they rated those on a 5-point, we'll call Likert scale. So underneath you can see they rated it based on not stressful, sometimes creates stress, all the way to so stressful, sometimes we feel we can't cope. And every study visit, they rated those same 5 outcomes on that same 5-point Likert scale. And so what you see on the right is the average score of those 5 items and how that changes over time. And so what we found here, and again, this is set up just like the last slide, where you've got treatment in the gray box, and observation for the 8 weeks before on the left and 8 weeks after on the right. And improvement is a decrease in the score. So again, we saw relative stability before treatment, improvement in this individual clinical response during treatment with Anakinra, and then once they go off, you see this rebound again of the average scores all going back up to being very similar to pre-treatment. Next slide, please. And another way to look at parenting stress that we really based that individual clinical response score on was this previously validated standardized measure called the Autism Parenting Stress Index. So this was developed for families with children with autism, and it asks the items that are listed here. So some similarities to what we asked in the individual clinical severity score, but this was developed specifically for autism. But since there was a lot of overlap in this, we thought this would be a good standardized survey to include as well. And here again, the parents or caregivers rated each of the items listed there on this 5-point Likert scale with the same idea of how stressful is each of these outcomes. And to the right are the data. And for some reason, the gray box didn't show up here, but it is, again, the treatment is from visit 0 or week 0 to week 36 with observation before and after, and improvement is a decrease. And so we see the same trend here, where there is here a bit of a trend of improvement before treatment, but then a statistically significant improvement by week 16 and week 36 while on Anakinra, and this rebound again once we stopped Anakinra. Next slide, please. Okay, now another way to really look at the heterogeneity or the diversity of the disease problems and manifestations is to use what's called the multi-domain responder index. And this was used in the MPS 7 enzyme replacement study drug development program, where they used an MDRI also. They had different outcomes, different items that they included in it. But the idea is that within a disease like Sanfilippo syndrome, MPS3 or MPS 7, you've got you have kids at different stages, and they have different things that they might benefit from with your treatment in one area, but not in another area, because maybe they're having more problems with movement and pain, but not having any problem with sleep at this point. And somebody else might be having a lot of problems with sleep, with behavior, and not with areas of pain. And so what this does is it allows you to show if your treatment is having benefit on multiple different items, instead of choosing just one and saying, okay, if it doesn't have a benefit for the majority on that one outcome, then we're going to say it's not effective. This is a nice way of being able to look at the variability in the disease and the variability in the treatment effects. So the outcomes we included here were different surveys that we used to measure movement disorder, fatigue, the parental stress, which was the autism parenting stress index I showed previously, pain, behavior, which was the Sanfilippo behavior rating scale, and sleep. And how these are set up, so we've got week 8, 16, 36, and in black, those are the number of patients who had an improvement on that item. And so for each of those things, the movement, fatigue, parental stress, we had a survey, and we had a minimal clinically important difference. Either it was published in another group of patients, or we used the 8 weeks prior to treatment to determine what one standard error, how much it would generally change without any intervention. And if the change over 8 weeks, 16 or 36 was greater than that minimal clinically important difference, then you would say they improved. If it went in the worst direction more than that, you say worsened. And if it didn't change more than that either way, then it's no change. And so what you can see here then is the number of patients at each time point who had an improvement in each of these different items. All right, next slide, thank you. So using that approach, just counting up how many outcomes people had an improvement in, we took that number, and I'm showing it here just at 16 weeks because this is what was included in the publication. So at 16 weeks, if you look at the number of multi-domain responder index, that the number of items they had improved on, and that is plotted on the bottom, the x-axis here, on the figures on the right. So going to the right up to 6, that would be the most number of improvements. 0 means they didn't improve on any of those items on the left. And we plotted that against what's called the immunophenotype. So Dr. Michelina Iacovino, who's going to join us in the Q&A and who is the lab colleague here at Lundquist with me, she did immunophenotyping on the patients as they were going through the study and looked at the number of, for example, what's shown here, CD4 cells, CD8 cells, monocytes, B cells, to see if those changed with treatment. And then using this method to see if the changes were associated or correlated with the clinical benefits we were seeing. And one of the reasons we did this was to provide a biochemical or biological confirmation of the changes that we were seeing clinically. And what we found was that what you see here is that an increase in the CD4 T cells by week 16 and a decrease in the CD8 T cells was associated with a greater number of improvements in these outcomes on the left, and a decrease in your monocytes is associated with a greater number of improvements. And so what that just generally is a less pro-inflammatory environment. So decreased pro-inflammatory environment is associated with the clinical benefits we are seeing. So that is what we would expect Anakinra to do. We did not see any association with changes in B cells. And Anakinra primarily targets more of the T cell side of your immune system. And there's overlap, of course, but B cells are much more related to your humoral immune system. And it is not thought to have much of an impact on your B cells. And so here we didn't see much change in B cells, and it didn't correlate with any of the clinical outcomes. So just a biologic confirmation of some of the benefits that we had seen. Next slide, please. And so since publishing these results, I've been getting emails and phone calls from some physicians and some caregivers and asking me some really specific questions about, well, we saw the data, my child or my patient is this in this stage of the disease or that, or having this problem. Do you think it would help them? And it's really hard, and I get why these questions are coming up, because it's hard to answer an individual patient question from a clinical trial like that has such a diversity in the population, and we have to, for the trial, really pool the data together. So what I've tried to do, and what I'm going to show here, these are published data that were just called post hoc, so this was not planned. I just tried to pull out some data to try to get a good idea of maybe how to answer some of these clinical questions. So the first one often I get is, well, what about disease severity? Did the patients with more severe disease, less severe disease, did you see any difference in how they responded to treatment? And so to try to get at that question, I used the receptive communication outcome, the GSV score on the Vineland. And this was based on work by Elsa Shapiro and Julie Isengard that found that specific measure on the Vineland, which we had collected, was a sensitive measure of decline in function in Sanfilippo syndrome. And so we thought, okay, that's a reasonable way to try to give a number to disease severity. And that was not the baseline Vineland receptive communication GSV score is not associated with changes over 36 weeks in any of the outcomes that I've shown. Next slide, please. And then I also looked at, okay, well, let's go at some of these specific problems. So what about sleep? What about pain? And if I took the baseline score for sleep or pain or parental stress, what I found was that, yes, the more problems the patients were having with sleep, the greater the improvement was in their sleep score. The more pain they were having at baseline, the larger change improvement they had on their pain score, the non-communicating children's pain checklist. And same with parental stress. The higher the parental stress was coming into the study, the greater the improvement during the study. And finally, the higher SBRS movement was associated with great improvements in movement. Next slide, please. And in terms of age at screening, age was not associated with changes in the Sanfilippo Behavior Rating Scale or any of the other clinical outcomes that I mentioned. So next slide, please. So to summarize, for safety, there's no new safety concerns. Anakinra there was no Anakinra related severe adverse events. For dosing, we determined that the 200 milligram daily subcutaneous dose is what's needed and is what we will be bringing forward to a phase 3 study. Next slide. And in terms of efficacy, we've seen improvements in the mood, anger, and aggression, social emotional dysfunction, movement and orality, and caregiver quality of life. We see variable improvements in pain, sleep, fatigue, and disordered movement. We see a change into a less prone family environment correlating with the clinical benefits we've seen. The benefits do not seem to be related to age or overall disease severity. However, those who have more sleep problems, pain, parental stress had the greatest improvements in those respective outcomes. Next slide, please. And I'll just finish by acknowledging and thanking the team that made this possible, both here at Lundquist, University of Minnesota, and a lot of thanks to Kara O'Neill and Glenn, both Kara for her scientific partnership on this, parental partnership, giving a perspective we wouldn't have otherwise had, and for the funding of the study from Cure Sanfilippo Foundation and Study Drug from SOBI. And a special thanks, of course, to the study participants and their families, because I know, and this meeting has made it even hit me more, and we really need to look at our protocols of how burdensome these trials are, and that we really need to do everything we can to make participation as easy as possible. So thank you.
All right. Hello, everybody. I wanted to first start by thanking Kara and everyone at Sanfilippo for having me here again this year. My name is Jonathan Acevedo, as Kara mentioned. I'm a PhD candidate at the Lundquist Institute at Harbor-UCLA Medical Center. Dr. Polgreen, who just spoke moments ago, is my mentor, and today I will be discussing the therapeutic potential of cannabidiol and Sanfilippo syndrome. So next slide, please. So I'll start by introducing cannabidiol. So cannabidiol or CBD, is a non-psychoactive compound that's found in the cannabis plant, and this is something that can be administered orally. CBD has been shown to be safe and effective in the treatment of children with certain types of seizure disorders, and this has been a formulation of CBD has been approved by the FDA, and I will touch on that a little bit earlier. But additionally, on top of the effects that it has on seizures, it also has some potential therapeutic benefits, including pain management, sleep disturbances, and behavioral problems, to name a few. These are just some that are quite relevant to Sanfilippo syndrome, but there are others that have been reported as well. Next slide, please. So getting into CBD and its therapeutic mechanisms, I know this slide there's a lot going on, but the main thing that I want you guys to take away from the slide is just that the mechanisms that CBD, the potential therapeutic mechanisms that CBD has, are very complex, and that CBD interacts with many different receptors and has different mechanisms by which it has these therapeutic effects. Just to name a few, I mean, as you can see in the top part of this graphic, it has anti-convulsant effects, anti-inflammatory effects, and antioxidant effects, which we believe would be relevant to Sanfilippo syndrome. And additionally, thanks to work that was done by a group in Italy, there has been work that has shown that in Sanfilippo syndrome, there is a dysregulation in the dopaminergic system and dopaminergic signaling. And there's been some work that has suggested that CBD has potential therapeutic role in the dopaminergic system. So for this reason, as well as the mechanisms that has been highlighted in this slide, we believe that it could be, there are many different targets that it could have therapeutic effects. So I want to highlight just one specific receptor or mechanism, and that's in one of the orange boxes, the GPR55. So for the next slide, I will be kind of zooming in on this specific mechanism. So next slide, please. So the GPR55 or G protein coupled receptor pathway is a pathway that is very important in many things in biology, but one of which that is relevant to us and the work that we are doing is that it's important in axonal formation and neuronal development. So some work that was done in Dr. Michelina Iacovino's lab, which Dr. Polgreen mentioned that she will be joining for the Q&A after to answer some questions. Some of the work that was done in her lab in certain proteomics testing was that we saw that there are many proteins that are dysregulated, especially upregulated in an MPS IIIB brain organoid model, where many of these proteins are involved in this G protein coupled receptor pathway. So this pathway is sort of primed to have a very large response, and a dysregulated pathway, especially when we're looking at neuronal development and axonal formation, can have many significant effects, especially as we will see here on the next slide, please. So as I mentioned, that G protein coupled receptor pathway is involved in neuronal development. So what we saw in a MPS 3A forebrain neuron model, cell model, which these cells were actually patient-derived iPSCs that were later differentiated into neurons, cortical neurons. And what we see is that in the healthy control cells, they have this healthy morphology where they have a very small cell body, long, thin axons or dendrites, certain branching. And then in the MPS 3A cells, we see this abnormal morphology where they have more of a short and wide shape to the neuron and a much larger cell body, which is demonstrated in the graphs down below, where the cell diameter, and this was just measuring the cell body, is significantly greater in the MPS 3A neurons. And the percentage of these abnormal cells, which we just defined as those with the larger morphology, the large, short and wide morphology, was significantly greater. So we can see, and one thing that I want to clarify is the control cells still have this quote unquote abnormal morphology, but this is just representative of a certain stage in neuronal development. This is very normal for cells to start out this way in this shorter, wider shape. And then as they continue to develop and become mature neurons, they will elongate and appear more as the cells do on the left. So this is just the takeaway that I want to just highlight here is the fact that these cells, these were imaged at the exact same time point. However, the MPS 3A cells appear more like the early developmental stage compared to the control cells. Next slide, please. And so we believe that this is related to the GPR55 pathway specifically. And on the little graphic on the left, we see that GPR55 is normally activated by LPI. This is just an endogenous or a molecule that occurs naturally in the body that activates this pathway. So in MPS 3, we anticipate that LPI would bind to this receptor and cause a very large signaling response that basically will produce this abnormal morphology in the MPS 3A neurons. However, CBD has been shown to be an antagonist of this pathway, and we believe that this will, we were looking at this pathway to see if it would correct that abnormal morphology in our MPS 3A cells. And what we saw, first starting with the healthy control cells, the graph on the left, was at this early stage in development, we measured the percentage of abnormal cells in just healthy control cells. So they didn't receive any sort of treatment. And that baseline was at about 40% abnormal cells. However, when when the cells were exposed to LPI, which again is that molecule that activates this pathway, we see a significant increase in the percentage of abnormal cells. However, when cells were exposed to CBD, so that pathway is essentially like blocked or inhibited, then we see a significant reduction in the percent of abnormal cells and a number that is very similar to, or essentially the same number that we see in the control. However, we can see that at the 0.5 micromolar dose of CBD, there was just an uptick in the percentage of abnormal cells, and that's something that we're still trying to better understand. So there wasn't quite that dose response that we were hoping to see, but nonetheless, still was significantly lower than those cells that received LPI. And so most importantly, in the MPS 3A cells, we see that at baseline, or the control cells, those that did not receive any sort of treatment, were at a much higher baseline in terms of what percentage of the cells were abnormal at about 60%. And similarly, when those cells were exposed to LPI, we see the percentage of abnormal cells up to about 80%, which is similar to what we've seen in the healthy controls. And then, just as we saw in the healthy control cells, we see that CBD significantly reduces the percentage of abnormal cells with a statistically significant reduction compared to controls in the 0.1 micromolar. And once again, we're seeing still a decrease in that percentage of abnormal cells in the higher dose. However, it's still a bit higher than the 0.1 dose. So that's something that we want to try to better understand, that we're actively testing in the lab. Next slide, please. So on top of just that specific mechanism, CBD has been shown to have anti-inflammatory effects. So in human studies, CBD has shown anti-inflammatory effects in humans by reducing the inflammatory cytokines, including VEGF, TNF-alpha, and IL-6. But more relevant to Sanfilippo syndrome, because this hasn't been tested in Sanfilippo, we also, again, Dr. Iacovino's lab, they measured the effects of CBD on MPS IIIB mice, and specifically in neuroinflammatory markers. So on the graph on the left, we see that CD68, which is a marker of microglia, sort of a neuronal, or sort of like a brain immune cell. We see that the MPS IIIB mice have significantly larger percentage of CD68 that are active in the brain compared to control mice. And then that's also the case with GFAP, which is just a marker of astrocytes, where we see a significantly larger amount of GFAP cells that are active in the MPS III mice compared to controls. And again, in the MPS III mice that received CBD, we saw a reduction in both CD68 and GFAP, so a decrease in the microglia and the astrocyte markers. And the astrocytes came down to almost the same level as controls. So that was just a kind of confirming our belief that CBD would be able to cross the blood-brain barrier and produce these anti-inflammatory effects in the brain as well as somatically. Next slide, please. So not only has CBD been shown to produce these therapeutic effects in like biological systems, but it's also been tested in a clinical setting. So in other populations, specifically because it hasn't yet been tested in Sanfilippo syndrome, we see that in autism spectrum disorder, there was a small study where 10 participants received a dose of 75 to 1, a ratio of 75 to 1 of CBD to THC. So it's CBD enriched cannabis over 6 months. And they showed that there were significant improvements in all of the symptoms that they measured, which is the graph on the right, which I will kind of walk through now. So starting with ADHD symptoms, they saw about a 30% improvement in the symptoms in this population. Behavioral disorders also saw about a 20% improvement in these symptoms. Same with motor deficits, they saw about a 20% improvement. The lowest amount of improvement that they saw was in autonomy deficits, but this was still about a 10% improvement compared to baseline. And then the communication and social interaction deficits saw about a 25-30% improvement. And then cognitive deficits also saw 20% improvement. But the largest improvements that were seen in this study specifically were in sleep disorders, where about 50% of the participants saw improvement in their, the participants saw about a 50% improvement in their symptoms. And this may be just related to the some of the side effects of CBD, where somnolence or like sleepiness is a common side effect that is seen in CBD. So that was not too much of a surprise, but definitely something that is very significant, especially in our population where sleep disorders are a big problem in Sanfilippo syndrome. Additionally, there was work that was investigating the effects of CBD in Fragile X syndrome. And this was, in this study, they were exposed to just CBD, no CBD to THC ratio here. And 3 months of CBD treatment improved all of the aberrant behavior subscales that they measured. Some of these include hyperactivity and irritability. These are just to name a few that are relevant to Sanfilippo syndrome. And lastly, Epidiolex, which is a pharmaceutical grade CBD, is currently FDA approved in pediatric populations for the treatment of seizures. And this is in specific populations such as Dravet syndrome and Lennox-Gastaut syndrome. Julie here will soon be talking about this. So I'm happy to, actually, sorry, I have one more slide before I pass it off to her. So this is why we are sort of turning towards Epidiolex or CBD for Sanfilippo syndrome. And part of the reason why we turned for this medication is because of the fact that during the Anakinra trial, we noticed that about 30% of participants were currently taking CBD, whether it be over the counter or if they were prescribed Epidiolex for certain seizure conditions. That's still unknown, but basically, since we had a decently large proportion of the participants in the Anakinra trial that are taking CBD, we want to make sure that we understand the dose that is required for if it has any sort of potential therapeutic effects, as well as just to test the safety of Epidiolex and Sanfilippo. And then additionally, just some of the therapeutic properties that are relevant to Sanfilippo that I highlighted in the presentation. That's because CBD has these properties, then we believe that it'll be a good candidate for a drug to test in Sanfilippo syndrome, as well as the fact that there's already proven safety and efficacy in other pediatric populations, however, in symptoms that are relevant to Sanfilippo. So I will now pass it off to Julie, who's a medical science liaison at Jazz Pharmaceuticals. And thank you.
Hi, everyone. So I'm going to talk to you about cannabinoids. And actually, if you could advance to the next slide, please. So I'm going to start with a quick overview of the FDA and the USP. So the FDA is the Food and Drug Administration, and it's the government agency responsible for ensuring the safety and effectiveness of foods, drugs, and supplements. So for medications, they require extensive testing to prove that they are safe, effective, and consistent before they reach the public. This includes cannabis products. Now, when it comes to ensuring quality and safety in medications, the U.S. Pharmacopoeia plays an important role alongside the FDA. The USP is a scientific organization that provides standards on what and how strong a medicine is and decides what information needs to be on the label. So you've seen this symbol before, this USP triangle. It should be on your vitamins. They have legally recognized standards for FDA-approved drugs and dietary supplements, which of course include all FDA-approved cannabinoid medicines. The non-FDA approved cannabis products do not have these legalized standards, so there's some confusion in the guidance on testing and consistency. Because of this, there is no real way to know exactly what is in each product. Okay, next slide, please. It's important to keep in mind that the cannabis plant is complex. The cannabis plant contains THC and CBD, which are molecules called cannabinoids. But cannabis has a lot more than just THC and CBD. It turns out that within the cannabis plant, there have been over 100 identified cannabinoids along with other molecules. So the key takeaway here is that most of these cannabinoids and other molecules have not been well studied. But THC and CBD do have an extensive research history. Next slide, please. So this is what I think is the most interesting part of cannabis. It is a very efficient bioaccumulator and phytoremediator. So a bioaccumulator is an organism, in this case a plant, that absorbs a substance quicker than it gets rid of it. So it absorbs it in faster than it metabolizes or eliminates it. Therefore, it builds up in the plant. A phytoremediator is a plant that's used to clean up environmental pollutants such as heavy metals or organic contaminants from soil, water, or air. These plants absorb, accumulate, and sometimes break down harmful substances, helping to restore polluted environments to a healthier state. In other words, cannabis is used to clean up toxins and other chemical waste from soil, water, and air by absorbing it into itself. Here's an interesting cannabis fact. After the Chernobyl disaster in 1986, cannabis was used as a phytoremediator to help decrease soil toxicity near the nuclear plant. So the key takeaway here is that cannabis is highly efficient at collecting heavy metals, toxins, and contaminants to the soil and air. All right, next slide. So what's stopping those contaminants from accumulating in products that people consume? Nothing. Any of these contaminants could be present in any of these products. So how much testing is done to ensure that the contaminants aren't being sold along with cannabis products? Testing is inconsistent and not mandated. So there's no guarantee that cannabis-based products do not contain contaminants unless they are FDA-approved cannabinoids. It's good to understand the different types of testing that need to be performed on cannabis-based products to ensure contaminant-free products. So there's biological testing. So this would test for types of toxic fungus. There's microbial testing, which would test for other pathogens like bacteria. There's heavy metal testing to look for contaminants that may have been in the water or soil that got absorbed into the plant. There's pesticide testing. If pesticides are used on cannabis plants, it's going to absorb them. And there's residual solvent testing. So solvents are used to extract those cannabinoids from the plant to put them into these products. And these solvents can be harmful. So you don't want to make sure that those solvents don't make it into the final product. And then there's potency testing. And so that would just make sure that what is on the label, the CBD or THC content that is listed on the label, is what is actually in the product. So another cannabis fact is that nickel was over 2.5 times higher in the leaves of cannabis plants when grown in toxic soil near a mine compared with a greenhouse growth conditions. Next slide, please. So this is a study by the Clean Label Project, and it shows labeling inconsistencies. So this study had 242 CBD products, and they were tested for heavy metals, pesticides, and other contaminants. First, let's talk about how the study results show inaccuracy of CBD products and also unexpected THC content. As a reminder, THC is the intoxicating component of the cannabis plant, the part that makes you feel high, whereas CBD is not intoxicating. Over 30% of products were inaccurate in CBD content. Moreover, even though none of the products indicated that they contained THC, 45% of the products had measurable levels of THC in them. But there's more. Not only was there a presence of THC in some THC-free labeled products, but some also had toxic elements, including heavy metals. The top 10 brands averaged 34 parts per billion of lead. To give you an understanding of this number, the action point of the FDA for drinking water is 15 parts per billion. So this is more than twice that in your top 10 CBD brands. I want to point out to you what the American Academy of Pediatrics says. There's no safe amount of lead exposure for children. Even small amounts of lead in their blood can harm their thinking, learning, and brain development. At low levels of exposure, the Centers for Disease Control and Prevention suggests getting kids tested and getting them checked and taking action. In short, any level of lead in a child's blood is a worry. There is no safe level. Next slide. So without FDA regulation, CBD products are highly variable. In short, it seems like products are consistently inconsistent. Levels of cannabinoids differ, and sometimes cannabinoids we aren't expecting are found in our products. A recent FDA report found that out of 147 products, more than half were mislabeled. They either had over or under the listed CBD content on the label, and that many of them contained THC when the label said that there was none. So not only was the cannabinoid content mislabeled, but again, toxic elements were found, including heavy metals, notably lead. Now, there's probably some good actors out there, but we know that there are some bad ones too. But we don't know who is who without the oversight of the FDA. So why do we care? Well, you probably want to know that what you're buying and administering to your loved one, and that you're not putting yourself or anyone else in harm's way. We also know that other cannabinoids can have effects on the body, and we want you and your healthcare provider to know exactly what you're taking. To complete this part of the presentation, I want to take a minute and tell you a story about a patient named Sam. Sam had epilepsy since he was 4. He suffered about 100 seizures per day. His family tried every available treatment and found no relief. After exhausting all options, they turned to cannabis products. The first tincture they tried reduced his seizures dramatically, from 10 to 20 seizures an hour to just 1 an hour. This was a huge breakthrough, a huge relief for Sam and his family. However, the next batch did not work, and his seizures returned in full force. When tested, they found that the tinctures claiming to have a 20-to-1 CBD to THC ratio actually had almost no CBD or THC. The lack of consistency and regulation made it impossible to rely on these products to manage his seizures effectively. Many other patients have had this exact same experience. So in conclusion, I hope this gives you a clearer understanding of the importance of consistency, regulation, and safety when it comes to cannabis-based products. Ultimately, we all want to make informed decisions that put the health and well-being of our loved ones first. Thank you.
All right, thank you for that, Julie. So for several of the reasons that have been highlighted, both in my talk and in Julie's talk, that's why we are turning towards Epidiolex and testing that on to see if Epidiolex has any sort of therapeutic effects on Sanfilippo syndrome. Specifically, we will be measuring the neurobehavioral and functional outcomes. This will be in a phase 1, 2, 3 clinical trial, where the first portion of the trial will be the sentinel safety cohort, where the first 5 participants will be screened, and then they will all be taking Epidiolex, and they will be titrating in a dose for the first 4 weeks. This will be done to test the tolerability as well as sort of a preliminary efficacy dose that will then be used in the subsequent cohort. So after 4 weeks of Epidiolex, then they will have the option, participants will have the option to participate in the open label extension, where they will have the opportunity to receive that maximally tolerated dose for 52 weeks. So in this trial design, which we are hoping to sort of just do it on one go, this sentinel safety cohort will sort of act as the phase 1 to 2 portion of the trial, where we're testing safety and tolerability. And then next slide, please. This second cohort will be the phase 3, and this will be a placebo-controlled cohort where participants will be screened, and then if they pass screening, they will be randomized into one of 2 groups, where they will, I'll direct your attention to the top line, for example. That will just be, we'll call it Group A. Group A will receive Epidiolex for 16 weeks at various doses. And at the same time, the other half of the participants that are randomized to placebo will receive placebo for 16 weeks. They will then have an 8-week washout period to ensure that CBD is completely out of their system, and then cross over to the opposite group. So group A will then receive placebo for 16 weeks. Group B will receive Epidiolex for 16 weeks at various doses. And then again, to test long-term efficacy as well as safety and tolerability, we will give all participants an opportunity to participate in the open label extension where they will all receive that maximum tolerated dose for an additional 52 weeks. And next slide, please. So I just wanted to highlight some of the key inclusion-exclusion criteria because I'm sure people will have questions, and I want to provide some information since we are hoping to get this trial going as soon as possible. Some of the exclusion or inclusion criteria will include, obviously, an MPS III diagnosis that is confirmed by genetic testing. We require that they are at least 4 years of age or greater, as well as if they are on either Anakinra, fluoxetine, or any probiotic supplement, that there are no dose changes in the last 8 weeks on any of these medications. And then for some of the key exclusion criteria, no participant can take any form of cannabis, including CBD, especially if it's over the counter, in the last 8 weeks. So that's one of the things that I want to highlight specifically for this talk, because if any participant who's interested in participating in this trial, then there's some time to sort of wean off of the cannabis product that they might be on between now and when we hopefully start enrolling soon. As well as no recreational or medical use of THC within the last 3 months. And then some of the concomitant use of therapies, including rifampin, diazepam, except for intermittent uses as needed as a rescue medication, or clobazam, valproate, and there are a few others, but these were just some of the more common ones that I wanted to highlight. And next slide, please. So I just want to say thank you to everyone. Again, I wanted to thank Cure Sanfilippo Foundation. And if anyone has any further questions or would like to know more about the trial, you can contact me at this email or the phone number listed down below. Thank you.
Thank you all. And I think we'll Dr. Iacovino, we're gonna see her, Glenn. There we are. Hello, Michelina. Hi. Thank you all for the wonderful talks and excellent information. Jonathan, we share your excitement to get going on the Epidiolex trial, and Cure Sanfilippo is happy to be providing the funding for that study, and thankful to Jazz for providing the prescription form of CBD called Epidiolex for the study. So we're excited to see that.
Yeah. Sorry, I just want to say not only thanking Cure Sanfilippo for the funding, but as well as the collaboration that we did to design this trial. So that was one of the things that I forgot to mention, was the fact that this trial was designed collaboratively, both with Cure Sanfilippo to get the family's perspective on what's most important, as well as FDA as well. So
thank you. So a question from the audience around the preliminary CBD data was just a clarification about the significance on some of the, I don't know if this was related to the GFAP CD68 slide or the other one around the GPR55 slide, but where you showed differences with treatment with CBD. Did those reach statistical significance?
Yeah, so I can touch on the G protein coupled receptor pathway that we did test and met statistical significance both in the size of the soma, like the cell body,
okay,
as well as the percentage of abnormal cells. The work that was done in the mouse model and MPS IIIB, that was done prior to my arrival. And I might have said statistical significance, but Michelina, could you touch on whether that reached that statistical? Okay.
I think you're on
you
mute.
might be muted, Michelina.
It did not reach statistical differences. That was just a small pilot experiment. It was coming out of the pandemic, and so we did not have a lot of animals. And most importantly, the animals were almost reaching the time we have to sacrifice. So it was just tiny pilot experiment, and we see a substantial decrease, but we need to repeat and increase the end of the animals to have clear statistical data.
Yeah, and I apologize for misspeaking on.
Yeah, with that trend, we have just to increase and repeat the experiment on with the larger numbers.
Thank you. Yeah, but that gave you an idea to, you know, hey, exactly. This may be a important direction.
We calculated the power needed to reach statistical, and we just needed barely to double the animals. So we use that as a pilot experiment to know what will be actual experiment to achieve a statistical difference.
Understand. Thank you. Thank you. Regarding the, well, let me ask it, is there any other information that we'd like to share on if patients are interested in either thinking about Anakinra or thinking about enrolling in the Epidiolex study, you know, how we might direct them? I know Jonathan provided some information about, you know, maybe drugs that they might consider whether they wanted to take or not, if they were going to be eligible, but any other kind of tips for families who might be interested?
Yeah, I think one, well, in terms of contacting us, Jonathan gave his contact information. Mine's out there. I'm happy to talk with anybody or connect through email about either of the studies. And but one of the additional comments I want to make that, you know, we've all as a team, and including you, Kara, have talked a lot about, was the fact that there's a placebo in it. And I know it's a really sensitive subject and something that that is understandably often not looked at positively. And I think it's important to make the point that the outcomes we're looking at are not cognitive. They're not the type of outcomes that are in the gene therapy, enzyme replacement therapy studies, where a placebo is, in my opinion, not necessary because of these really concrete outcomes, objective outcomes that you're looking at. The Anakinra study, the Epidiolex study, because of the stage of disease that we are targeting, and it's really symptom management, hopefully, you know, decreasing the rate of progression of disease. But for now, it's symptom management, but it's caregiver, parent reported outcomes. They're more, we call it subjective. So knowing you're getting a treatment can potentially make you answer in a more positive way. And there's a lot of data out there in other areas to support that. And so for that reason, just to explain, you know, to everybody that we did include a placebo. We are including a placebo in the phase 3 Anakinra study because the outcomes we're looking at are just more likely to be influenced by the placebo effect than something like cognition.
Yeah. And in the design of this study, however, it is a crossover. And so all patients enrolled will have the opportunity to receive the active drug, and the period of time in which they are on inactive drug is really short. And so I know that was something that we spent a lot of time thinking about. What was the shortest amount of time that we could use to get a conclusive answer, and really being mindful of limiting that amount of time. So it was 4 months, right?
Yeah, and we built in an interim analysis. So, you know, there's a one time we check when everyone gets to that first placebo-controlled period to see, can we take out the placebo? If we reach our endpoint, then we can take everybody off placebo at that point. So we added that in to try to decrease placebo time, as well as the 52 weeks. You know, like if you do this for just 4 months, 1 year of open label. That was something that was really important to us. And we're so thankful to Jazz for agreeing to, because that's a big ask for a pharmaceutical company to give another whole 1 year extension. And so that, I think, is also an important part of it. And we'll have that, hopefully, and it looks like we'll have that in the phase 3 study for Anakinra also.
Yeah, excellent, excellent. Well, I'm excited to create these opportunities for patients to try a treatment that could help their symptoms, but also answer some really important questions scientifically for our community going forward. And thank you all so much for being here and for the work that you're doing and commitment to our community.
Thank you.
Thank you.
Bye, everybody.